Hello everyone. The next topic is about the central dogma model of the molecular biology. So what is this model is about? So this model is about the genetic information that is from the DNA to RNA to protein. So the process of the central dogma model is basically showing the transcription and the translation process. So this model is basically showing the two types of processes. So it is defined as a process in which the information in DNA is being converted into a functional product. So it is suggested that the information that is being present in the DNA is essential to make up all the proteins, and RNA acts as a messenger that carries the information through the ribosomes. So this is about the central dogma model of the molecular biology. Now first we will come to understand the definition of this. So it is a process in which the genetic information flows from the DNA to RNA to make a functional protein. So for example, this is a DNA. There is a transcription. There is RNA, and then the translation occurs and it will convert into the protein. So this is about the whole mechanism. So reverse transcription, there is also a reverse transcription, and there is a process which is called as replication, in which the DNA will make its own copy. So it will make its own copy. So that is called as the replication. Now, what is central dogma model? So the central dogma illustrates the flow of the genetic information in the cells, the DNA replication, and the coding for the RNA through the process of the transcription, and further RNA codes for protein by the process called as the translation. So this concept of a sequence of interaction can be understood through the framework. So what framework we are talking about here is the most common includes the biopolymers, and the major category of biopolymers includes the proteins. So RNA and DNA that are being further divided into general transfers, unknown transfers, and special transfers. So special transfers occurs in an exceptional case in the laboratory. So transfer almost in general transfer occur almost in all the types of the cells. So it describes the regular flow of information through the transcription and the process of the translation. And unknown transfers, they are said never to occur. So the new strand, a DNA strand, they are formed with one strand of the parent DNA and the other is newly synthesized. So this process is also called as the semi-conservative DNA replication. So this process is known as the semi-conservative process of the DNA replication. Now what are the steps that occurs in the central dogma model? So first of all, one strand of the new DNA is the parent DNA, and the newly formed is the newly one is synthesized. So this is a semi-conservative DNA replication, and this the transcription translation are the central dogma model. So meaning of the central dogma model comes from both the processes, that is the transcription and the translation. So now coming to the steps. So what are the steps that are being involved in the central dogma model? So there are basically two steps that are being involved in the central dogma model, that is the transcription and the translation. So transcription is about when the DNA gets converted to RNA, and translation process is about so when the RNA gets converted to the protein. So this is about the steps that are involved in the central dogma model. Now, first is about the transcription. So what is transcription? It is the process by which the information is being transferred from one strand of the DNA to the RNA by the enzyme. So which enzyme is used here? So the enzyme used is RNA polymerase, because here DNA is getting converted to RNA. So the enzyme used will be the RNA polymerase only. So the DNA strand which undergoes the process consists of three parts. What are the three parts here? So the three parts are the promoter, structural gene, and the last is the terminator. So these are the three parts in which the DNA strand undergoes these three processes. So the DNA strand that synthesizes the RNA is called the template strand, and the other one is called as the coding strand. So the DNA dependent RNA polymerase, it binds to the promoter and it catalyzes the polymerization in the 3' to the 5' direction. As it approaches the terminator sequence, which is the last part, the terminator part. So as it reaches the terminator sequence, it terminates and releases the newly synthesized RNA strand. So the newly released RNA strand further undergoes post-translational modifications. So the next is about the process of the translation. So in the process of the translation, what is happening when the RNA is getting converted into the proteins? So it is the process by which the RNA codes for specific proteins. So it is an active process which requires the energy. So this energy is being provided by the charged tRNA molecules. So ribosomes, they initiate the translation process. The ribosome consists of a larger subunit and a smaller subunit. The larger the subunit in turn consists of two tRNA molecules placed close enough so that the peptide bonds can be formed at the expense of the enough energy. So mRNA enters the smaller subunit, which is then held by the tRNA. So tRNA here describes that it is a transfer RNA molecules of the complementary codon present in the larger subunit. So there are two subunits which are present. One is the larger subunit and the other one is the smaller subunit. So the two codons, they are held by tRNA molecules placed close to each other and a peptide bond is formed between them. So as this process repeats, there are long peptide chains of amino acids which are being synthesized. So this is what is about the translation process in which the RNA gets converted into the protein. So this diagram is only depicting that the RNA is converted to protein. So the RNA moves, the ribosomes, it moves along the RNA reading the genetic information, and as the ribosome, they reach the gene, amino acids are assembled as per the information in the gene, and these amino acids bond together in the ribosome, moves along the RNA strand to even naturally form the complete protein. So here a protein, individual amino acids bonded together. So here the protein has been formed from the RNA. So yes, this was all about the central dogma model of molecular biology. So these are some of the references. Thank you.